would like to thank the FDMAS Alliance and also the organizing committee, specifically Mike and Natasha, for putting together such a wonderful program and for giving me the opportunity to present our work. So I guess for this audience, I don't have to talk about or repeat what McCune-Albright syndrome or FD is. I just want to say that my lab studies bone or skeletal biology and diseases, and the long journey of our work on FD and McCune-Albright syndrome dated way back when I was at NIH with a close collaboration with Mike and Pam Roby. I'm glad this collaboration has been quite fruitful, and we are still working on it. And the work is spearheaded by a very talented postdoc, John Regard, who is now a medical director at Novartis in Boston. And the reason why we are interested in McCune-Albright syndrome is twofold. Of course, we want to understand the cellular and the molecular mechanism underlying this devastating disease. And we, from the science point of view, at that time, we want to understand how Wnt beta-catenin signaling is transduced as a rare disease. Tell us a lot about this. And it was known at that time that G-alpha-S is involved in transducing Wnt beta-catenin signaling, but the data were controversial. Since lots of the work has been published a long time ago, I would just say that, and again, this was collaborated with Mike, we find a distinct alteration in FD or McCune-Albright syndrome patient stroma cells in the marrow is the upregulation of Wnt beta-catenin signaling. And to make a very long story short, what we find is that the gain-of-function mutation in G-alpha-S leads to upregulated Wnt beta-catenin signaling activity and this sustained high Wnt beta-catenin activity blocked maturation of osteoblasts when they differentiate from mesenchymal progenitor cells or stem cells. So we proved this in vitro in tissue culture, and with the cells that Mike gave us from either healthy individuals' marrow stroma cells or those cells from McCune-Albright syndrome patients. And why, if we coach these cells to differentiate as osteoblasts, the healthy cells will form normal bone nodules. After von Casa staining, they will have this characteristic marble-like pattern, but with the mutation in it, and we don't see this marble-like pattern. But if we knock down beta-catenin, and there was a progressive rescue of that nice marble-like pattern. So reduction of beta-catenin will allow us to form better and more mature osteoblasts, and therefore rescue the phenotype possibly. So encouraged by this nice result, we decided to generate another mouse model of McCune-Albright syndrome or FD in mice. So what we decided to do is to put this mutation back in the original locus or the endogenous locus of GNAS. What we did is shown here. So we put a wild type minigene in front of the exon where the MAS mutation was found, and we also knock into this mutation. So after, without Cre-induced recombination, this wild-type minigene will just express a wild-type G-alpha-S from this locus, so the mice will be normal. After Cre-induced excision of this minigene, and then the wild-type minigene will be deleted, and then at the same time, the mutant allele will be expressed from its endogenous locus. So this, we believe, allows us to most precisely model this disease in a tissue and cell-specific manner, as long as you have a Cree tool. So the first thing we did is to show that if we activate this mutant expression in the germline that causes heterozygous early embryonic lethality, we use this SOX2 Cree, which is active in ovocyte. As you can see here, the mutant embryo was already very small at E10.5, and by E12.5, it is almost dead. So this explains why this disease can only be uncovered in human as mosaic, because if you inherit this as a germline, it will kill the embryo. So then we decided to express this mutant allele in osteoblast-specific lineage using the Ostrix Cree, and as you can see here, it mimic what has been found in human and in other mouse models. There's bone overgrowth, and there was increased osteoclast activity, and moreover, and also the sort of loss of the trabecular bone, and then everything becomes loss of cortical bone. Everything becomes trabecular bone-like thing. So we find, and there's a marrow fibrous change. And again, we prove this is at least partially due to upregulated Wnt beta-catenin signaling, because if we genetically remove one copy of the Wnt receptor LRP6, and that is sufficient to largely reduce the mutant phenotype, and moreover, we proved this again in an in vitro assay. We used the adenovirus Cree to induce the expression of this mutant allele in the marrow stroma cells, and after osteoblast differentiation, as you can see here, and this reduced osteoblast ossification shown by von Casa staining, if we treat this culture with an inhibitor of Wnt secretion, and there is a dose-dependent rescue or enhancement of osteoblast differentiation of the mutant cells. So this really says that upregulated Wnt beta-catenin signaling is at least one important reason why these cells cannot mature and cannot mineralize very well in the patients. So because this lesion oftentimes occur as a genetic mosaic, so we decide to model that more precisely in adult mice using a tamoxifen-inducible allele. Again, this is a skeletal. This is quite specific to the skeleton system using the STOX9 Cree ER line. And we label the mutant cells with tamoxifen. So the red cells are mutant cells and the rest are wild type cells. And as you can see here, if we express this mutant allele in the tamoxifen, in the TD tomato, a positive red fluorescent cells, first of all, there's increase in the TD tomato positive cells. So that is consistent with the previous observation. There's increased expansion or proliferation of the mutant cells. And second of all, we find increased expression of early osteoblast marker Ostrix. However, if we look at the expression of osteopunctin, which mark mature osteoblasts, and their expression was reduced. So again, that proved this lesion is due to a blockage of osteoblast maturation. And more importantly, we find that there is a profound non-cell autonomous effect of the mutant cells. So one of the hallmarks of this disease is marrow fibrosis. So we used alpha SMA to mark those fibrotic cells. As you can see in the wild-type control, and there's very few alpha SMA positive cells. They are mostly found in the smooth muscle cell layer outside of the blood vessel. However, in the mutant, you see a lot of alpha SMA positive cells, and many of these alpha SMA positive cells are not TD tomato positive, namely, they are not mutant cells, they are wild type cells, but they are influenced by the mutant cells to become fibrotic. And we heard a lot about the craniofacial problems of FDMAS that has been very well documented in the literature. And we find in the craniofacial area, again, if this mutant allele is expressed and there is upregulated Wnt beta-catenin signaling, here we use exon 2 LAC-Z as an in vivo readout for Wnt beta-catenin signaling. If you see these blues staining are Wnt beta-catenin Wnt beta-catenin activities, and you see more blue, there's more Wnt beta-catenin activity. So this FD or McCune-Albright syndrome mutation did increase Wnt beta-catenin activities, and that was further confirmed by RT-PCR. And furthermore, we used pharmacological inhibitors of Wnt beta-catenin signaling pathway, and find that can be used to rescue the defect to a certain extent. With all this said, I would like to talk about a little bit of the Hippo signaling pathway, and that we have recently connected with FDMAS. So why Hippo signaling? First of all, like Wnt beta-catenin signaling, this pathway is also involved in the regulation of osteoblast proliferation and maturation. And moreover, the transcription factor YAPTAZ, like beta-catenin for Wnt beta-catenin signaling, mediate the transcription activation of the signaling pathway, critically regulates rancoligand that is produced from osteoblast. If you lose YAPTAZ, and you will see a huge upregulation of rancoligand, and that was known. So, and upstream of YAPTAZ, there is a cascade called Hippo signaling or Hippo kinase cascade. And basically what it does is to regulate the phosphorylation of YAPTAZ transcription factor. And once they are phosphorylated, like beta-catenin, they will be sequestered in the cytosol and then degraded. If they are not phosphorylated, meaning if the upstream kinase were not activated, and they will accumulate and then migrate to the nucleus to activate downstream gene expression. And one of the readout that we commonly use and other people in the field also use is CTGF and the CYR61 expression. And why, apart from this, and the reason why we want to focus on this pathway is that in vitro experiment already showed that GFS through pKa can regulate LATS phosphorylation, and then this phosphorylation will activate LATS and therefore increase YAPTAZ activity. So theoretically, if there is an activation of GFS in the mutant cells, LATS will be more activated and YAPTAZ will be more phosphorylated, and therefore their activity or protein levels will be reduced. So we use the same mouse model and to test that hypothesis. And as you can see here, as we have previously shown in this model, Ostrix-Cree-driven GNAS mutant model, while Wnt beta-catenin signaling was activated, now we see a reduction of YAP protein level, both based on immunohistochemistry and also confirmed by Western blot. So the YAP protein was reduced, and then its downstream target, CTGF expression, was also reduced. So there is a reduction of YAP transcription or protein activity. And we also have, in the beginning, I thought I only have 10 minutes because I have to give 10 minutes to Mark. But if we genetically express activated form of YAPTAZ, we can promote bone formation, maturation, and all these good things. So To summarize, what we want to test is this G-alpha-S activation may also regulate osteoblast proliferation and maturation by regulating YAP-TAZ. And we have some preliminary data in collaboration with Mark Wine that sought induced kinase 2 and 3 is also involved, critically involved in this process, and Mark has assumed that if you delete SIK2/3 and that generates a phenotype very similar to G-alpha-S activation. Mark's supposed to talk about his work on SIK2/3, but due to the weather, he couldn't make it. Anyway, so hopefully next time we have more data to share about the entire pathway in FDMAS. And with this said, I would like to thank current and previous trainees in the lab who are instrumental for the work, and our collaborators, Mike, Pam, and Mark, and our funding agents. I would say thanks to FDMAS Alliance and through Penn $1 million bike ride program, and we now got funded by DOD to study FD in more detail.
Thank you, Yingzi. Let's take a question or so for Yingzi. Does anyone have questions for Yingzi? Eris? No? Sorry. Over here.
Actually, lots of questions. That's so beautiful, your work. A very basic one. Sometimes I notice you have CRE driven by SOX9, sometimes it's Osterix, sometimes it's PRX1. Does it make a big difference in the phenotype, in the overall mouse? Is there a reason to change the driver, or could it have been? So I understand the need of doing something kind of skeletal specific, but is there an important difference between those, or would they all give more or less the same phenotype?
That's a great question. So as a geneticist, of course, there are compelling reasons for us to choose different CREs. So PRX1 is the earliest one that is active in stem cell progenitor cells, and they are sort of very early on in embryos in all the cells that will go to future long bone lineage, for example. So with that said, you are going to see the most striking defect, but you lose the opportunity to study the cell non-autonomous effect, namely how the mutant cells affect the surrounding wild-type cells. And that can only be accomplished by SOX9 CRE-ER, so because it's inducible. If you don't inject tamoxifen, the mice will be normal. So we can wait until they are adult, and then we can inject tamoxifen, and we can inject tamoxifen with different dosage. So you can titrate down different size or different number of mutant cells and see the effect. So why Osterix? Osterix is further down the lineage. It's already committed to osteoblast. So is this mutation only generate a phenotype in the progenitor or differentiated cells? To basic scientists is also important. So that's why we use the Osterix CRE to activate the mutant allele expression. But with all this said, overall, if you look at the lesion, they are about the same.